DNA replication. DNA replication is really a fascinating process that occurs within every single cell, and it is really, actually, it's a complex process. I'm not going to lie, it's a complex process. Your book explains it in such a way that is oversimplified, but we will teach you in the way that is oversimplified because that's what your test is going to test you on. But before we talk about DNA replication, I just want to get into some brief minor characteristics of DNA that we've previously talked about, and I'm going to introduce some new ideas about DNA, but now we're going to finally start understanding the entire process. So hopefully you find this somewhat interesting, or at least you will finally find it interesting at the end when we finally do translation. So let's go back to DNA. Deoxyribonucleic acid, deoxyribonucleic acid. And let's talk about it. Deoxyribonucleic acid. It's a double-stranded structure. I'm going to label it as DS. DS means double-stranded. Double-stranded structure. We have a heel-shaped structure, right? It's intertwined, it's twisted. It's like a twisted ladder, as some scientists have described this DNA. DNA also happens to be anti-parallel. These DNA strands move in an anti-parallel direction. What does that mean, anti-parallel? It's a very important concept to understand. Anti-parallel. Meaning that one strand of DNA runs this way, and the other, the complementary strand, runs the other way. This is called the anti-parallel direction. Very, 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 very important when we start to learn about labeling DNA. You're going to see in just a few minutes. So we have a double-stranded structure, we have a double helix, and an anti-parallel. It goes in the anti-parallel direction. The thing about DNA is that DNA replicates in a semi-conservative fashion. Semi-conservative fashion. And I'm not going to explain this right now because after we learn about replication, then I'm going to show you what semi-conservative really means. It's going to be very easy for you to pick up what it means. So just keep these characteristics in mind. And now let's talk about DNA in further detail. So DNA is composed of nucleotides. They're composed of nucleotides. DNA is composed of nucleotides. And what's a nucleotide? Nucleotides contain three things. Nucleotides contain three things. Contains a 5-carbon sugar. In this case, what is my 5-carbon sugar? It's my deoxyribose. Deoxyribose. If I was talking about RNA, my 5-carbon sugar would be just ribose. RNA stands for what? Ribonucleic acid. So I have my 5-carbon sugar, which is my deoxyribose. I have a phosphate group. And lastly, I have my nitrogenous bases, or one of my nitrogenous bases. And if you remember from the previous video, I have 4 nitrogenous bases that I've taught you so far, and they are called adenine, thymine, cytosine, and guanine. So, I want to draw this out actually because it's going to help us understand in a better manner. So, here's my 5-carbon sugar. Here's my antagon. Here's my 5-carbon sugar, and it's connected to a phosphate group. We'll label it as a P. And well, actually, let me redraw this actually. Put the phosphate here. Doing it for a specific reason. I'm going to label this as my 5' strand. So, I have my phosphate group right here, I have my 5-carbon sugar right here, and attached to it is a nitrogenous base. So you can pick any base in this case. We're just going to pretend it's the base C. Cytosine. Here is a nucleotide, a phosphate group, 5-carbon sugar, and a nitrogenous base. let's continue, because it's just not one nucleotide does not make a DNA, it's multiple. So let's continue and say, okay, and we can continue and continue and continue. I won't continue on for much longer. And let's choose all the bases. Let's pretend this right here is A, this is G, and this is going to be T. Here is a single strand of my DNA, 5-carbon sugar. So, 5-carbon sugar, phosphate group, nitrogenous base, phosphate group, 5-carbon sugar, nitrogenous base, and so on and so on and so on. This is just a single strand, and now I'm going to label this strand. I'm going to label, you see where this phosphate group right here is? I'm going to label this phosphate group beginning with 5'. We're going to call this a 5' region. And you see this right here, this 5-carbon sugar? This sugar right here we're going to call a 3' end. So this DNA strand is running from the 5' to 3' direction. This DNA strand is running from the 5' to 3' direction. 5', 3'. Students get confused about this so many times. We're going to make a separate video explaining the 5' and 3' structure. But put it this way. If we're starting off with the phosphate group, it's 5', and if we're left with the sugar at the end of the drawing, it's a 3'. And usually, I always draw a structure. I mean, anytime I draw DNA, it's always from the 5' to 3'. I always draw DNAs from 5' to 3' because it makes my life easier, and I would imagine that it makes every other biologist's life easier as well. So here's a single strand, but DNA is double-stranded. Well, erase it, but DNA is double-stranded. So now I have to draw the opposite side. However, it's anti-parallel. So what does that mean? So instead of 5' to 3', now 5' to 3' is going to go from here to here. Okay, so 5' to 3', 5' to 3', and now let's draw this. So, I'm going to draw a phosphate group over here. Going to draw 5-carbon sugar. Now you might be saying, hey, why is this drawn backwards? It looks very weird. Well, it's in the anti-parallel direction, so technically the phosphate, if it was anti-parallel, it should be reversed. Your textbooks usually have it written the way you would normally see it, but it avoids confusion. But I just want to be a tiny bit accurate here. And, well, I've already messed up this drawing, but let's connect this all the way down here and say, okay, what binds to T? Well, A binds to T. And A and T, they'll nicely fit together. Well, they don't really fit together. There's some kind of hydrogen bonding between them. These bases, how do A and T connect together? How does C and G connect together? How do they form that complementary double strand? How do they form that double strand? It's due to hydrogen bonding. This A and T and C and G have specific hydrogen bonding to them. So A and T actually have 2 hydrogen bonds between them. I'm just going to draw 2 dotted lines to represent hydrogen bonding. So every single A and T have 3 hydrogen bonding between them, and C and G actually have 3 hydrogen bonds between them. So let me finish off this drawing. Well, I'll have a backwards phosphate with a 5-carbon sugar. Connected. I know this diagram is very messy, but I'm just trying to prove a very simple point. Here's cytosine, and this cytosine is going to be connected with 3 hydrogen bonds, and so on, and so on, and so on. You get the entire picture. I just want to stop real quick and say that the G and C don't actually have 3 hydrogen bonding between them. If you look at the most scientific literature, you'll find that this DC bond is stronger in hydrogen bonding due to stacking energy. Now, this is a whole different concept. Apparently, it's a bit complex for students to interpret. Frankly, I don't think it's too hard, but they just teach it to you as G and C have 3 hydrogen bonds between them, and A and T have 2 hydrogen bonds. So, I will teach you that A and T have 2 hydrogen bonds between them, and C and G have 3 hydrogen bonds between them. Remember that. It might show up on your exam. It probably will show up on your exam. So please, just have a mental note of this diagram. Once again, here's a nucleotide. Here's several nucleotides. It makes up a DNA strand, 5' to 3'. On the opposite direction, I'll have 5' to 3'. This explains, or this shows, the anti-parallel direction of DNA. I'm going to erase all this, and now get into some more complex stuff, like replication. Now, if I drew this and try to explain replication to you, we would be here for hours and hours, because for starters, I can't draw, and secondly, it would look very, very messy. So let's talk about replication, and we're going to oversimplify this picture so we can understand the basics. First things first is replicate. Let's talk about replication. Why do you want to replicate DNA? Why do we have to replicate DNA? Well, how many cells do we have in our body? Remember, we've already learned about mitosis and meiosis in the previous videos. Right now we're in molecular genetics. Remember the cell cycle? Remember the cell cycle here? I'm not going to go into too much detail, but over here somewhere you have the S phase, which stands for synthesis. This is the phase, if you can recall, where DNA is replicated. Right? And the S phase is in the interphase stage. Remember, the S phase is in the interphase stage, and DNA will be replicated there. It's going to double its mass in order to ultimately mitotically self-divide, and of course have an entire new copy of the DNA, so it can structure proper proteins. So, first thing to remember is, where does replication occur? At the interphase stage, specifically the S phase. But now we're going to learn, how does replication occur? And you know what? Replication is somewhat tricky. It's not a nice thing to learn, but I'm going to try to make it as easy as possible for you. So stay tuned for part 3.